The overall goal of this microinjection protocol is to modify the western corn rootworm genome.
This method can help answer key questions regarding western corn rootworm biology and their ability to quickly overcome pest control methods such as Bt toxin and oral RNAi.
Besides western corn rootworms, this method can also be applied to similar species, such as southern and northern rootworms.
I first had the idea for this method when I was asked to help with injection-based RNAi in western corn rootworm back in the 90s.
Generally, individuals new to this method will struggle because rearing western corn rootworms in the laboratory is labor-intensive and survival rates remain low using current technology.
Obtain between 500 and 1,000 non-diaposing rootworm adults from a reliable company or research laboratory and house them in 30 cubic centimeter cages. Store the standard rootworm artificial diet in food storage containers about 1 centimeter deep. Unused diet can be stored at 4 degrees Celsius for up to 2 months. To feed the rootworm, load 100 centimeter petri dishes with 10 to 15 grams of diet. Load 1 dish of food into each rootworm cage and replenish it when the supply runs low or the diet dries out. Also keep a 300 milliliter water reservoir covered by a cotton ball in each cage to serve as a water source. Change the water when it runs low. Maintain the caged rootworm at 26 degrees Celsius with 60% humidity and a 1410 light cycle in an insect rearing incubator. Set the lights off time later in the day, such as at midnight, so eggs are laid before